Hello again, everyone. Welcome back to this second day of the APMEN University online course on insecticide susceptibility monitoring. Yesterday, we had tutorials 1 and 2, and today we will look at tutorials 3 and 4, and these being on the CDC bottle bioassay method and the cone bioassay. Each of the two videos is only about 8 minutes long, somewhere around there. But at the end of each tutorial, we will have a short poll. And I want to ask each of you, please, to fill in the poll question, as it really helps us to understand what we are doing right, what we are doing wrong, and how we can improve to give you a better product. So at the end of both tutorials, we will have a Q&A session where you can pose whatever questions you would like to ask about the tutorials. Please feel free to post chats and comments and write to your friends listening in the chat box. But please put your questions in the Q&A box, please. We will not, it just gets too confusing to try and monitor different boxes. So we will not extract questions from the chat box. We will only look at questions from the Q&A box. To respond to your questions, we are lucky again to have from Insecticide University, Dr. Jiro Nararak, who was with us yesterday, but also Dr. Rachadawan, lots of experience, and also Dr. Chutipong Sukanon, all three from Department of Entomology at Insecticide University. So sincere thank you to those colleagues. Unfortunately, Professor Terapap is tied up with other commitments, and so he's not able to help with the Q&A session today. If you have watched all 4 tutorials and you're interested in getting a certificate of attendance, then we will issue you with a certificate, but you need to complete a short multiple choice questionnaire to at least show that you've listened to all 4 tutorials and you were concentrating fairly well. You need to score at least 70% out of the 10 questions, but they're fairly easy questions if you were listening. Right at the very end, and also, so I'm going to suggest, please can I ask you to get pencils and a piece of paper ready, because in a few seconds time, I'm going to ask Wulan, our technical officer here, to show you on screen how to get to the multiple choice questionnaire. So we're going to show you where you can access the multiple choice questionnaire, and we ask that you complete this by 11 a.m. on 1st of October. That's day after tomorrow. So you've got the whole day tomorrow, and then we're going to close the site after 11 a.m. on the 1st of October, 11 a.m. Bangkok time. And then we'll send you your certificates to the successful candidates. It'll be signed by myself and Professor Terapap, the certificates. So Wulan, can I ask you just to put on screen that link site so our audience members can just write down the URL. Wulan will also post it in the chat box, and Wulan will also email that link to everyone that's listening in today and also yesterday. Wulan will send you that link so you can access the multiple choice questionnaire. It's 10 questions. Just fill it in. It's little boxes. You tick the correct or the wrong one. Please read the question carefully, because sometimes you need to answer the wrong, you have to indicate the wrong answer. And sometimes you must tick the right answer. So you need to read the questions carefully. Just so you are aware, these sessions are being recorded and will be uploaded on the Aurene website, https://aurene.org. So you can watch them again anytime or refer your friends to them. Then a reminder that this APMEN Insecticide University online course is made possible by funding from the University of California and also the USA Centers for Disease Control and Prevention. And then I'd like to thank the team that quietly in the background makes this possible, makes this presentation possible. So my thanks go to Dr. Tin Cho Thu, who's our APMEN VCWG program coordinator in Bangkok, Dr. Ponsia Hein, who is the APMEN program coordinator in Singapore, and then Mrs. Shobicha Wulandari, everybody knows as Wulan, who's technical officer for Malaria Consortium in Bangkok. Okay, so let's go to the first of the two tutorials today. The first one is about the CDC bottle bioassay. So I'd like to mention that like the WHO paper tube bioassay, the CDC bottle assay is also used to check for the presence of insecticide resistance in a mosquito population. So it's also a tool to check for resistance. And basically, a bottle, he will show you in the video, a bottle is coated with a known amount of insecticide, the diagnostic dose. Mosquitoes are then put into the bottle and observed for 2 hours. Resistance is then determined by the percentage of mosquitoes that die, the mortality rate, at the predetermined threshold time during those 2 hours. The test should be run for the entire 2 hours unless all mosquitoes have died earlier than the 2 hours. Okay, Wulan, can you put on the video for us, please? Thank you. Hello everyone. In this video, we demonstrate the CDC bottle bioassay. The CDC bottle bioassay was developed by the United States Centers for Disease Control and Prevention, shortened to CDC. It is also a widely used and internationally accepted surveillance tool for detecting resistance to insecticides in vector populations. The CDC bottle bioassay relies on time-based mortality data, which are measures of the time it takes an insecticide to penetrate a vector, traverse its intervening tissues, get to the target site, and act on that site. The following are the main materials or items required for the assay. 250 milliliter glass bottles with plastic screw-on lids. 1 milliliter graduated disposable pipettes, aspirators to transfer mosquitoes, the mosquitoes themselves, mosquito holding cups, amber-colored or foil-wrapped bottles for stock solutions, a timer, permanent marker pens, disposable gloves, paper sheets, pens and pencils for data recording. So we start by coating the bottles as follows. You need to have made sure that all bottles are clean and uncontaminated, and you do that by using test mosquitoes to check on mortality. So first, check that all the bottles and caps are completely dry. Add 1 milliliter of acetone into each of the 2 control bottles and put the caps back on tightly. Prepare the insecticide solution by mixing the technical insecticide compound with acetone solvent according to a ratio that you need to look up in the CDC guidelines, which we will give you at the end of this video. Add 1 milliliter of the insecticide solution into each of the 4 treatment bottles and put the caps back on tightly. After having added the solutions to the bottles, swirl the contents inside so that the bottom is coated. Invert the bottle and swirl to coat the inside of the cap. Place the bottle on its side for a moment to let the contents pool. Rotate while rocking the bottle gently so that the sides all the way around are coated. Repeat this for all the test bottles. Allow the bottles to dry for 60 minutes. The bioassay can be performed with the bottles either in an upright position or with the bottles lying on their sides, but you have to be consistent. If you start with the bottles in an upright position, keep doing so. Next, introduce 10 to 25 mosquitoes into each of the control and treatment bottles. Start a timer. Be sure to examine the bottles at time 0 and count the number of dead or live mosquitoes. If you find dead mosquitoes at time 0, make a note of them on the form. Record how many mosquitoes are knocked down or responsive, whichever is easier to count, every 15 minutes until either all are dead or up to 2 hours. You do not have to go beyond 2 hours. Record these data on the reporting form. Observe mortality in the control bottle at 2 hours and record in the results form. Then transfer the mosquitoes back into the holding cups. Note again that mosquitoes are considered dead or knocked down if they can no longer stand. Assess the mortality of mosquitoes in the control bottle after 2 hours. If mortality is between 3 to 10%, correct all mortality figures using Abbott's formula. If mortality is greater than 10%, then discard the results. You will have to redo the assay. You will have received an Excel spreadsheet with your CDC bottle kit, and this spreadsheet will automatically plot a time mortality curve as you insert your data. Assess your results to determine if resistance is absent, partially present, or fully present. Finally, clean the bottles. Wash the bottles with warm soapy water and rinse thoroughly with water at least 3 times. Then rinse the bottles and the caps with acetone. Let the bottles dry. Then, to ensure that the cleaning procedure is adequate, introduce some susceptible mosquitoes into the cleaned bottles. If there is higher mortality than in a control group in a known clean container after 2 hours, then repeat the washing and drying. You can read more about the CDC bottle bioassay at the following URLs.
Sorry, you're on mute, Leo.
There we are. Thank you, Wulan. Thank you, Tin. So yes, apologies for that brief glitch in the video presentation. Got a bit nervous there, but fortunately it didn't last very long and it wasn't a very serious one. We'll try and work out what happened there. So I think, Wulan, if you wouldn't mind just putting on the short poll, and I'm going to ask everyone if you can just answer. It's very few. I think it's about 5 or 6 little questions, straightforward. If you wouldn't mind just responding to the little poll there to help us understand if we've kind of done okay or we need to improve or what. I'm going to keep quiet while you fill in the poll questions. Thank you for that. think when we get to 80% of participants responding, we can start again. Gerard, I'm not sure if you saw that comment there about the 25 seconds or so on. Think about it in the meantime, and maybe you can respond to that comment, because I'm not sure what it was. I may have been distracted a bit. I'm not sure. Maybe just have a look at that comment and maybe respond to it. Okay, we're on 83% response. Thank you, everyone. There was a question or comment here about, can we get a link to the tutorial? Yes, I think Wulan, you did show the links. Maybe at some stage, again, you can put up the, or maybe just pop it in the chat box, the link again, where participants can access the link that will be, where it will be posted within the next 24 hours. Thank you, Wulan. So, okay. So I just want to make this comment before we go on to the next video. So I'd like to emphasize what I pointed out yesterday. The idea is not that if you watch this video, you'll now be an instant expert in knowing how to conduct the test. I think it's important to realize that you need to download the CDC bottle bioassay manual. And you can go to Google and just type in the CDC bottle bioassay, click, and it will give you, it's easy, it's straightforward. CDC bottle bioassay manual or guidelines provided by CDC. There's a link for that. And then you need to do the two in conjunction. It's a very straightforward, simple manual, a few pages. Just read that. And the value of the video is that sometimes you read what it says in a written text manual, but you don't quite understand what they mean by it. And that's where the video becomes very helpful. So between the two of them, they help each other. It makes it easier for you to interpret what to do, kind of reinforces each other. And it's wonderful. I promise you, for those of you who haven't done it, it's wonderful being able to do these insecticide resistance assays. It's one of the, if not the most important, it's one of the most important things for a malaria entomologist in a national malaria control program to be able to do, is to be able to detect and monitor the development of insecticide resistance. Your whole malaria control program can fall flat if you are not aware, not conscious, that your bed nets are no longer working, no longer efficient, your indoor residual spraying or whatever. Vector control is largely based on insecticides, the use of insecticides, and you need to know how susceptible your vectors are to those insecticides. So being able to do these assays is critically important. So the second tutorial that we're going to move to now is on the use of the cone bioassay. So the cone bioassay is completely different to the WHO tube assay and the CDC bioassay. It does not test for insecticide resistance. What it does is to see if the active ingredient in insecticide is still working after an amount of time has passed in a treated surface. So for example, if you sprayed the wall of your house with an insecticide in May 2021, then you can place some mosquitoes in a cone against that treated wall 3 months later, or whenever you want to see if the insecticide is still killing mosquitoes, or if the insecticide is no longer effective. And the same with a bed net. If you buy your own untreated bed net and you dip it in a bath of insecticide, then 6 months or a year later, you can test that net by exposing mosquitoes in a cone against that net to see if the mosquitoes are still being killed. So the cone bioassay is to test for residual efficacy of insecticide in or on a treated surface. In fact, you don't even use wild mosquitoes usually. You use known insectary specimens that you know are fully susceptible and have zero insecticide resistance in them. And you put these fully susceptible mosquitoes inside the cone and expose them to the treated surface. So Wulan, can we move to the next video, please? Tutorial 4. Hello everyone. In this tutorial, we are going to discuss and demonstrate the WHO cone bioassay. This is not a test for insecticide resistance in mosquitoes, but instead a test for residual efficacy of insecticides in insecticide-treated surfaces. Is the insecticide still working, or has it decayed significantly? So really, we are not evaluating the mosquitoes so much as evaluating the treated surface. WHO cone tests were originally designed to evaluate the toxicity of insecticide-treated bed nets against malaria mosquitoes. But cone tests are also perfectly suited to investigate the toxicity of other impregnated textile surfaces, such as impregnated clothes or insecticide-sprayed walls. In this video, we are going to use insecticide-treated bed nets, but the principles are exactly the same, whatever treated surface you want to test. The following are the main materials or items you will need for the assay: labels, mosquitoes, mechanical aspirator, plastic cones, treated net, green, control net, white, 70% ethanol, cotton wool, control slide, treatment slide, 2-ring rubber bands and 3-ring rubber bands. Moving to preparation of mosquitoes, we use adult female Anopheles mosquitoes of a known fully insecticide susceptible strain, all the females being 3 to 5 days old. 5 mosquitoes are required for each replication and 10 series of assay repetitions are recommended. So in total, at least 50 mosquitoes are used per net. No sugar starvation is necessary, but mosquitoes should not have fed on blood before being used for these assays. You will need at least 100 female mosquitoes, all between 3 and 5 days old, all non-blood fed, and all from an insecticide susceptible strain. 50% of these are for testing on insecticide treated material, and the other 50 are untreated control material. You need to label 10 cones as test cones and 10 cones as control cones. You will be putting 5 mosquitoes inside each cone, so a total of 100 mosquitoes. So step 1, you mark 10 cups as control, numbered from 1 to 10. So for example, C1 for control, cone number 1, C2, C3, up to C10. And then you mark the other 10 cups as test cones, or cups, also with replicate numbers, such as T1, T2, and up to T10. If there is more than one net to be tested, make sure you adapt the coding so that you can track which net is being tested. Clean the treatment and control slides with 70% ethanol. This is the control net. Attach the plastic cone to the net like this. Close the open hole of the cone with cotton wool and do the same with all the cones. Confirm that the mechanical aspirator is working with charged batteries. Aspirate 5 female mosquitoes into the tube. Carefully blow the mosquitoes into the cone and block the hole with cotton wool again. Check the time. You need to expose these mosquitoes for only 3 minutes inside the cone. Then aspirate the mosquitoes back into the cup. Now repeat the same procedure with the remaining control cones until you have completed a total of 10 control cones, each having 5 female mosquitoes. While you have been doing the tests, make sure you have spaced the cones in a way that you get good representative coverage of the net or the wall or whatever surface is being tested. Now repeat the entire exercise for the treatment or test cones on the treated material. Place a wad of cotton wool soaked with 10% sugar solution on top of each cup for the mosquitoes to replenish energy. 1 hour later, record the number of knockdown mosquitoes in each cup into a pre-prepared data sheet. Then place the holding cups in an area free from any predators, such as ants inside the insectary. 24 hours later, record the dead or moribund mosquitoes. Finally, clean all the equipment with an appropriate detergent or solvent. Please note that bioassays should be carried out at 27 plus or minus 2 degrees centigrade and 75% plus or minus 10% relative humidity. As a reminder, here is Abbott's formula again, shown on the screen. And then you can read up more on this assay by going to the following sites. Wonderful, excellent. So, can I ask you just to pop up that poll again for this particular video, if you don't mind? So to our audience members, I'd be very grateful if you can just fill in this poll as well, and then we'll move to question and answer session. Please feel free to answer questions. There don't seem to be many questions, or Girard and Carly have been responding to them very well in writing. Okay, sorry, I've lost the tutorial now. I'm assuming that, most people have responded. Thank you for that. So then we can move to questions and answers, and there are very few, or in fact, there are now none left. So let's give it a few seconds if anybody wants to ask more questions. What are the major advantages of the CDC bottle assay compared to WHO bioassay? So, Dr. Chutipong, do you want to try that one?
I think that because the CDC bottom assay, you have to look at the time as well, because you have to also determine the diagnostic dose and the diagnostic time, right? So it depends on the region when you establish the dose and time. Some places they use 60 minutes, some places they use 15 minutes. So I think because the WHO tube bioassay, they use the fixed time. And from what I have researched, I think that the WHO, I'm sorry, the CDC bioassay can be used to determine I'm sorry, I need to formulate my thought. It can be used to determine how the mosquito become more resistant over time. I would say like this. Yeah.
You mean the intensity of resistance, Chutipong?
Yes.
Yeah. Whereas with the WHO tube test, you have to use different papers with starting off with double the recommended dose, and then 5 times the recommended dose, and then 10 times so it's fixed steps.
Yes.
Whereas with the CDC bottle bioassay, you get a graph that plots, the mortality over time. So you get a more finely graded idea of the intensity of resistance in that population. Is that correct?
Yes, that exactly.
Yeah. All right. And my understanding also is that the CDC bottle bioassay is less expensive than most other techniques. So that's a big advantage as well, just the cost factor.
Yes.
And also, I don't know about you, but I've had problems in the past. If I want to get a WHO paper, a tube test, it's a challenge to order, and you have to wait quite a while. Plus it's expensive. I shouldn't be saying this, but so sometimes it is a bit of a problem with the WHO test kit getting hold of that. So there are advantages, quite aside from the technical efficiency of the two methods. There are other aspects to it as well. Jiro, do you want to add anything to that comment? You're on mute, Jiro.
Okay, and also one thing that we should consider is about the chemical. Also, they have some research that I see that some chemical is like, because the WHO contain assay that we treat the chemical on the paper, and for the CDC bottle assay that we coated in the blood, so that it would be have the effect to expose this mosquito, that some chemical they are like dissolved, or they are better than on the paper surface or the glass surface.
Okay, so I see Dr. Pradeep Srivastava, our very esteemed and respected colleague from India, our previous co-chair of the Vector Control Working Group, has joined us. Thank you very much. Thank you for taking time to join us, Dr. Pradeep. Dr. Pradeep, as a previous director in National Malaria Control Programs in India, may I ask if you have any opinion about the preference, say your own response to which do you prefer using, the WHO tube test or the CDC bottle bioassay? Do you have a personal preference, or how do you feel? You're on mute, by the way. You're on mute.
Am I audible now?
Yes, you are. Thank you. No, you're back on mute. Okay.
Thank you, Leo. See, basically all the 3 things which you have been telling since yesterday, they have got different roles in program mode. When we do resistance test through tube test, it gives first-hand impression that the population is resistant or susceptible to some extent in different criteria. But in case you want to take a decision for change of the insecticide, The bottle assay gives the intensity that what level of resistance is there, which is usually, and at the moment, it is not being done by and large by the program people. It has been available and limited to the researchers only. So we can always get the data feedback from them. Because this bottle assay was not available earlier, we used to do epidemiological impact was plus the results of the tube test on insecticide resistance. So if epidemiological impact you do not want to have that, you can go for bottle assay, and then you can see that 5 times of this insecticide or 10 times of this insecticide concentration. If you confirm that yes, 100% you are going to have the development resistance, then you can go on taking a decision. This is very important. Regarding the cone bioassay, there was one question that how can you compare? There is no question of comparison with cone bioassay with resistance. One should not try also. That is only for the toxicity at different interval in the field, whatever insecticide we have sprayed, the claim which is actually that. So for program personnel, I would say that that much knowledge is very, very enough, and it has been very scientifically explained in your videos.
Thank you, Dr. Pradeep. Wonderful. So while we have you available, before you cut off again like you did yesterday, let me ask you before you disappear possibly. So there was a question yesterday and I responded by saying at least once a year, but let's get your opinion. The question was, how frequently should one test for development or the possible presence of insecticide resistance? I said at least once a year. What's your opinion?
This is basically $1 million question. Technically, we should do twice. Depends on your epidemiological total scenario, the persistent malaria transmission, or it is a seasonal transmission. So you are right, absolutely, that minimum once in a year, because it depends on how many sentinel sites and what is the resources available for you. Then the another challenge comes nowadays. We are also facing in India also that some of the species like minimus or nowadays my complex that balabacensis or tyrus, adequate number what is written in WHO manual that 20 to 25 mosquitoes per replicate, it's not available. So that these are the challenges. You can't have twice, thrice or 4 times. So minimum once is absolutely correct, your answer. But if you can get at least pre-monsoon and post-monsoon, twice would be good enough to generate the data.
All right. Dr. Pradeep, thank you so much for coming in. We appreciate your participation. Thank you for that. So there's a question here from Rahul who says, how many times can we reuse the same bottle? Girard, do you want to give us a response to that question?
Yes, I think Dr. Srinivong already typed it on. So for the bottle, the bottle bioassay that can reuse is it should be like 3 or 4 times, and they can keep it for months, but you have to keep in like in a good condition. I mean, don't against the sunrise.
All right. Thank you,
Yeah.
Girard. Then there's this question by O Singh. What are the major advantage of CDC bottle bioassay compared to WHO tube bioassay? We've basically answered that already, and I think we pointed out that the WHO tube assay is good at least at giving you an initial idea, is there resistance present in this population or not? And if there is, then you can move to the 5 times or the 10 times the dosages to give you a kind of a stepwise indication of the intensity of resistance. Whereas the CDC bottle bioassay is more finely graded and you can plot mortality along the graph and it will give you a better indication of a more finely graded extent of resistance intensity in the population. So Ezekiel, you say, I have 2 questions on cone bioassay, but you don't tell us what they are. So maybe you can give us those questions. Jaipal, what is the impact on the result if we include Travis female mosquito on cone bioassay test? I'm afraid I don't understand that question. If a member of our panel understands that question, maybe you can try and answer. Mohammed Khalilur Rahman asked, can I determine the resistance intensification through the CDC bottle bioassay? Yes, you can. I think we just explained that you monitor mortality or knockdown over a 2-hour period, and you plot that on a time-based graph, and it will show you the intensity of resistance. It's not Travid, it's Gravid, Jaipal. Where was your question now? You see the questions jump around and I lose my place on this. But you said, what is the impact on the result if we include gravid female mosquitoes on the cone bioassay test? Who would like to try and respond to this question? Dr. Rachadawan, do you want to give it a try? Or anyone else, doesn't matter, anyone jump in.
Okay. Sorry, it means they are like, what is the impact result if we use the gravid female, right?
Gravid female in the cone bioassay test, yeah. Or it doesn't matter, yeah. Okay.
Let's imagine for the cone test that we test the mosquito exposed to the bed net. So that is not rely in the field. If you realize, because like the bed net that we use to protect the mosquito, that they got protect the mosquito bite to the human, right? So it's probably yet before it means that the mosquito that would be tested is could be unfit, because it's like they have to again the because we use the minute to protect the mosquito. Even the mosquito already fit and gravid, so they don't land on the bed net. So it means like they are like not like, how do you say, like it's a real thing that we use like the gravid mosquito to test with the cone test assay.
Yeah. All right.
Yeah.
Gerard, I'm sorry. Maybe my audio isn't working very well. I struggle to hear you a little bit, but my interpretation is that it's best to try and standardize always on adult females that are 3 to 5 days old that have not been blood fed, so that you get a standard response across all specimens in the population to exposure to the test surface. The problem is, if you start bringing in blood-fed females or gravid females or older females, they have different physiological responses to a challenge. So, let's say, for example, you want to test my fitness level, but I quickly go and drink a bottle of Red Bull or something. I'm going to perform better in that fitness test because I had access to additional nutrients. And so maybe a female that has a blood meal will give a different physiological response to being tested against a particular insecticide. Enough to influence, just increase survival over a period. Sorry, there's somebody talking. Can I ask you to mute your... Yeah, there we are. So it's best to try and standardize within the test population. So here's a question. Is the CDC bottle bioassay a substitute for the WHO tube bioassay? So I think we've already kind of answered it between all the other questions, that both will test for presence of insecticide, but the tube bioassay is more to give you an initial idea. Yes, insecticide resistance is present or not, and then it can also give you a big stepwise 5 times the normal dose or 10 times the normal dose, whereas the CDC bottle bioassay gives you a more finely graded idea of the intensity of resistance. But you can use both of them to test for presence of insecticide resistance. So yes, you can use the CDC bottle bioassay for in resistance intensity. Adrian, you asked, what do we do if the selected household spray with active ingredient bifrenthrin for the test wall cone bioassay was disinfected because the household owner probably died from COVID-19? Complicated for me to. Jiro, do you want to answer?
Sorry, I don't know how to.
I know. What to do if the selected household spray with active ingredient for the test was disinfected? Oh, okay. So if the house was sprayed with whatever insecticide, doesn't matter what it was, but it was disinfected, then you can't really expect the cone bioassay to give you an accurate response. You can do the test and it will tell you whether there's sufficient active ingredients still being exposed for mosquitoes to be come into contact with. It will probably be that the disinfectant will have impacted on the efficacy of the active ingredient, but the cone bioassay will give you some idea still if there's still residual efficacy to impact on mosquito populations. So I think, yes, do it, test it. It will give you an idea of the residual efficacy of the insecticide, even if you have disinfected the wall. You know, there should still be some insecticide being released, and the cone bioassay will give you an idea. We seem to have lost all the questions now.
Hello, Dr. Leo. We have a question from Dr. Shanta Kumar in chat box. I think this is about CDC bottle assay. Hello.
Okay, you say it's in the chat box.
Yeah. So from Dr. Shanta Kumar.
Chat.
Wulan, could you please read out the question, please?
Yeah, please, Wulan.
You're on mute. Sorry.
Oh, sorry. So yeah, Dr. Shanta Kumar asks, I think this is about the tutorial 3 CDC bottle say. Hello, I see that it's mentioned 10 to 25, but we need to precisely tell that evenness number to be maintained across the bottles. Otherwise, it will lead to confusion, right?
Thank you, Wulan, and thank you for the question. So if I understand correctly, it relates to the number of mosquitoes. Tell me if I'm wrong. You can put it in the chat box. Tell me if I'm responding to if I'm not interpreting the question correctly. I think you're saying 10 to 25 mosquitoes per tube or bottle. And that we should rather be exact and say, no, it must be 20 or 25. It doesn't actually matter. as long as statistically the sample size is adequate to give you a valid statistical proportion. You're not using the absolute numbers. You are using the percentage or the proportion of mortality. And therefore, it doesn't matter if it's any number between 10 and 25. It doesn't have to be standardized on exactly 20 mosquitoes or whatever, because you're not using actual numbers, you're using the percentage. I'm not sure if that makes sense. Jirod, would you agree with that response, or do you have a different response?
Yes, I agree with this one. With your answer.
All right. So I'm going back to the Q&A box. So the last one at the bottom there is, why should we have to use only 5 mosquitoes in each cone? So Dr. Chittipong, it looks like you're busy typing an answer there. Do you want to respond to that question?
Yeah, I think because the cone is quite small and we have to make sure that because the WHO cone bioassay is a contact assay, we have to make sure that all the mosquito inside is contact with the treating surface. So if we put too much mosquito, they can disturb each other and fly away from the contact, right?
Yes. Yeah, very good response. That's a good response. Thank you, Dr. Chittipong. From Zahid, in the cone test, some insecticide-treated surfaces show repellent effects on the test mosquitoes. The flying behavior increases among the mosquitoes. Does it reduce the exposure effect? Yes, that's exactly the reason that Dr. Chittipong was referred to. That is why you try and optimize the number of mosquitoes so that the disturbance effect is reduced, and at least, you know, you can have some control. At least you're not putting in too many for the mosquitoes to disturb each other, and therefore maximize period when the mosquitoes are going to sit quietly on the exposed surface. But if the insecticide has repellent properties, I agree. But then, you know, whether it's in the cone test situation, or whether it is in the natural situation where, you know, you're testing you're putting out an LLIN, an insecticide-treated bed net, whether it's by testing using the cone or the natural population, both sets of mosquitoes will be repelled. So ultimately, you are testing the same effect. And I think that's been kind of built into the cone assay as well. So I understand what you're getting at, and yes, it is a very good question, that, but you know, I don't know what other test you're going to be able to do to overcome that repellent effect. It's a very good question, but I think that you know, the cone test has shown itself to give you a fairly reliable answer in the tests that have been used over a number of years now. So despite that repellent effect, it can still give you a good idea of the residual insecticidal effect of the treated surface. So the question here from Srishanta, can we use mosquitoes to test or control while previous day use as control? So Jirod, I think, can we use the same mosquitoes for control purposes on different days?
It means, can we use the mosquito to test or control while
let's say the laboratory has very limited number of mosquitoes available in the insectary. Can they reuse the same mosquitoes from the previous day? My general response.
In the control population, right?
Exactly.
Is like the ideally is I do not recommend, because like for the control and the treatment, it should be the same condition so that we can compare.
No. Yes, you're right. You're right.
Yeah. So I agree with both Jirod and Dr. Pradeep that, I mean, if you don't have an option, then you don't have any choice. But where possible, always go for new, fresh mosquitoes. So you're not using specimens that you've already used and disturbed and so on. So if at all possible, use fresh mosquitoes for every replicate. Okay, I think we've run out of questions. I don't see any new questions. In which case, have I forgotten anything? I think we were.
Hello. I think in the chat box from Giovanni Harishra. May I know the question?
Sorry, here's one. Okay, I'll go to the chat box in a moment, Wulan. But here's one more question that came here from Jimmy Isamache. How do I know if mosquitoes were 2 or 3 days old and not blood fed yet? So that is why we use known population specimens, so that you collect wild mosquitoes, you take each individual female, you put it in a tube with a few drops of water at the bottom and some filter paper. You wait for that female to lay eggs, and then you use the larvae that come from those eggs. You put that in their own water bath, and then you take the female that laid the eggs and you do morphological identification and PCR identification if need be, so that you know precisely what species those offspring, the larvae, what species they are, because you've now gone and done a PCR on the female that laid the eggs. And because you are now rearing the adults that will emerge from those larvae, you know exactly when they emerged. You're keeping them in the insectary. You know exactly how many days old they are, and you've made sure that they have not received a blood meal. So the specimens you use are usually insectary reared specimens, F1 generation, first offspring generation from wild caught females and so on. So it's within your control to know exactly how old are those females and what is their feeding status. Okay, let's go to chat box and have a look here. In the field, as it would be difficult to identify the age of mosquitoes. So what should we do in the case of wild population mosquitoes? Or do we do cone? So or Dr. Rajdawan or Dr. Chittipong, please come in here. But my experience has been, I never use wild females directly. I take those females, blood feed them, put them in individual tubes, take the eggs, let the larvae hatch, keep each batch of larvae in its own tub of water, do a PCR assay on the female so I know what species it is, and then I can mix larvae of the known same species together, let the adult females emerge, and those are the female mosquitoes I would use in my insecticide tests, whether it be the cone, whether it be for WHO tube test or CDC bottle assay. For the cone bioassay, you use known, fully susceptible strains of. So Girod, would you agree with that?
May I supplement something?
Yes, please, Dr. Pradeep.
What you are saying is very nice, ideal condition. If there is a good research facility and a smaller area and a program or country like India, sometimes you do not get so much facility and allowing them and then getting. This way, even in WHO manual also it is written. If you do not have the good insectary facility to rear the mosquito for getting equal age to get rid of all those experimental errors. So we can use the field caught mosquito. The only problem is the blood fed mosquito in the morning when you collect, allow them for 4 or 5 hours to take rest. When they are semi-gravid, you can start doing your experimentation. So we have been doing and hundreds and hundreds of experiments. We have been doing in the afternoon putting, then after 30 minutes, and then next day 24 hours after we used to take this readings, keep taking the mosquitoes along with us after the exposure tube. So in field condition, it can be allowed just to get the, because in district level or sub-district level, it will be very difficult. Even selection of mosquito itself is a big problem. But yes, ideally, we should allow the mosquitoes to bring to the lab, do the egg laying, then get the same age mosquitoes and then do the exposure.
Thank you, Dr. Pradeep. Very practical response there. Sometimes it is difficult. Sorry, there's somebody. Can you mute your microphone, please? So sometimes it is difficult. Sometimes you want a quick answer. The problem there is, you know, you have the problem that you don't always then know exactly what is the species identity of the females that you tested. You know, you probably now down to species complex or species group level, but you don't really know exactly which species you are dealing with. So it becomes a little bit of a crude, I would say it gives you a crude indication of what's going on if you follow that method of using wild-caught females to do a field test of insecticide resistance. Use it as an initial indicator, and if you can, take some wild-caught females back to the lab and do it more thoroughly, the way I described earlier on, by getting F1 generation offspring, where you can do full checks on species identity, etc., and have them all at the same physiological status of age and blood-fed stage, etc. So, but for initial guidance in the field as to whether there may be resistance present, yes, by all means, you can use wild-caught females and try it out. It gives you an initial indication. So in the CDC bottle assay, if the susceptible mosquitoes are not available for standardization of diagnostic dose and time, how will we do this for a particular region? Anyone on the panel, would you like to respond to that? In the CDC bottle assay, if the susceptible mosquitoes are not available for the standardization of diagnostic dose and time. So I'm not sure if I understand this question correctly. The whole point is, if you are using field specimens, you don't know whether they are susceptible or resistant. That's the whole point of using the bottle bioassays or the tube bioassay. is to determine if the wild specimens you caught in the field, if they are susceptible or if they are resistant. So I'm not sure if I understand this. It's easier to respond in terms of the cone bioassay, in which case you need to use usually laboratory reared or insectary reared specimens from a population that is known or proven to be fully insecticide susceptible. So I think we've reached our time limit. Wulan Tin, is there anything you would like to add that I should be saying or you would like to add? Otherwise,
No, it's just actually we still have one question in the chat box.
in the chat box.
Yeah, this is from Giovanni. So Giovanni asks, when cone test is done for a wool sprayed with a residual insecticide, can blood fed female used? I'm not sure if I.
Your voice broke off, Wulan. So could you repeat that question?
Yes. So when contest is done for a wool sprayed with a residual insecticide, can blood-fed females can we use the blood-fed females? I think that's the question.
Okay. So as a general rule, we try and go for females to standardize. It's all about trying to standardize. So always, as a matter of general principle, always try and go for specimens that are 3 to 5 days old, that have never had a blood meal. Then at least you get a standard response across all the specimens that you are testing. If you don't have any other specimens, by all means, then use whatever specimens you have available. But then you must know that, you know, you're not getting an absolutely perfect response. And as long as, you know, it's just 1 or 2 specimens that are blood fed, whereas the others are not blood fed, I think you can get away with it. So, you know, if you can't avoid it, by all means, go ahead, but try and standardize as much as you can. Gerard, response from you?
Yeah, I agree. And also, like, I would like to respond some question that you are explaining before about the field population that we use. That if we cannot identify the age of mosquito, it means like when we test the mosquito in the field, it's so very hard to exactly the condition of mosquito. And also that it ideally that we would bring the mosquito to the lab and then to make the lay egg, use the F1 the offspring of mosquito. But in some case of not free, some species is very hard to be led in the field. So it's necessary to like early test can be tested only in the field. It means it's very hard to letting them to be a F1 and test it in the laboratory. So what we do, so like we like relate that our result to the control. So similar, we put them together and we see that because we have the control, but the result is supposed like it's not perfectly, but we can see the estimate the resistance of mosquito in that field. I mean, it explains for like my in some case of like about the physical condition of mosquito if they are not suitable for like realization or assay.
Okay, thank you. This is going to be our last question. And it says, in field caught mosquitoes, there's a problem of getting the required species and to identifying them and separating them from other species at the spot. How to carry out the I presume, hello?
I think they ask like how to carry out the experiment in this kind of situation, right? If we can use the mixed species or not. For
Okay.
this question, yes,
Do you want
I
to
don't
try and
think that.
answer it? Okay,
Yeah.
please, Dr. Rajdawan.
In this question, I don't think we can use the mixed species in the contest or in the bottle assays, because the first of all, I mean, different species they present a different mechanism to respond to the chemical, and the second point, you have to concern about how big the population, because when you perform the experiment, you have to perform the data analysis as well to confirm your result. So for this question, I would say no, we cannot use the mixed species in, I mean, in the same condition or the same cone or the same bottle assays.
I fully agree with you. Thank you for that good answer, Dr. Rajdawan. So just to elaborate there, if I may, very briefly, but we're going to stop here now, is that, you know, if you use 10 randomly caught mosquitoes of mixed species and you use them in a tube or CDC bottle bioassay, and the thing is, you want to try and establish the resistance or susceptibility status of the malaria vectors. They are the ones that you are interested in. You are not interested in the Culex or Aedes or even the other Anopheles species that don't transmit malaria. So now if you have 10 specimens, some of them are Culex, some of them are Aedes, and some of them are other Anopheles, but they're not malaria vectors, those will not generally have been exposed to insecticide, and therefore they will be susceptible. So now you get a result, because you put them all together, a result that suggests that the population is fully susceptible. But, you know, you may have only had one malaria vector in that sample of 10. And that one didn't die. And that's the one you really want to know about. If that one that didn't die is resistant, you want to know about that one. But because it's less than 98% mortality, you think the population is susceptible. So it's not good to use mixed assemblages of specimens. So I'm afraid we need to end it off there. And I want to express my sincere gratitude to the audience for your interest, the lively interaction, the questions you posed. I hope you've enjoyed it as much as I did, as I'm sure the rest of our panel did. It's been a privilege and a pleasure to interact with you, the audience. Thank you for your interest. And I want to thank Dr. Jirod, Dr. Rajdawan, Dr. Chutipong. Thank you so much for your time, for making yourselves available to help us to answer some of the questions from our vector control community in Asia Pacific. So to Tin and Dr. Pon and Wulan also, thank you for your support and help. Thank you all. Have a wonderful day. And thank you again. Khop khun krap. Thank you.
Thanks, everybody.
Thank you very
Thank you.
much.